O-Level Biology 5090, Chapter 17, Inheritance, Part 1. And in this Part 1, we will discuss about variation and DNA. So what are variations? Variations are actually the differences in characteristics among individuals of the same species. And some of these variations are passed on from the parents to their offsprings via genes at the time of reproduction. And some of these variations are resulted when there is change in the surroundings or in the environment or what an individual does. So the variations are of two types: continuous variations and discontinuous variations. So what are continuous variations? The different characteristics within a population. So the type of variations which are continuously originate generation after generation. And the examples can be given as height, body, mass, intelligence. So the height, body, mass, and intelligence are the continuous type of variations which are originated in all generations. So the height in the first generation can be different from the individuals of the second generation. So the second generation can have the difference in the height in the individuals of the third generation. So these are the type of continuous variations, and the discontinuous variations are those variations which have distinct features, and there is no variations, you can say, generation after generation. So they limited type of variations are there. As for example, the blood groups, only four types of blood groups are available, that is A, B, and O. So there is no other type of blood group which can appear in different generations. So four types of blood groups are available there, and then other examples are tongue roll and earlobe. So in this way, we can say that these are the types of discontinuous variations. Discontinuous variations deal with a few clear-cut phenotypes. Few clear-cut phenotype. As I have told you that only A, B, AB, and O type of blood groups are available. There is no other blood group. And then deals with spectrum of phenotypes ranging from one extreme to the other. As for example, height. So height will be changed in every generation. So there can be many different uh, differences in the height generation after generation. So the discontinuous variations are controlled by few genes, whereas the continuous variations are controlled by many genes. So because they are the type of continuous variations, and genes do not show additive effect, but the in continuous variations, the genes show additive effect. That is, at the same time, the many genes can express themselves. As for example, the more dark genes, the darker will be the skin color. So in this way, they show the additive effect in the continuous variations. Not modified by environmental changes. Environmental changes does not affect the discontinuous variations, whereas the continuous variations are affected by the environmental conditions or change in the environment. For example, greater exposure of the skin to sunlight will produce a darker skin color. So skin color can be changed or other problems can be produced in these types of continuous variations. So both of these types are different from one another. Now the next topic is DNA, deoxyribonucleic acid. And deoxyribonucleic acid is made up of two strands which are twisted together. So these two strands of DNA, they are twisted together in this way. So a shape known as double helix. So double helical is formed by the twisting of these two strands of DNA. Each strand has a backbone made up of alternating sugar and phosphate groups. You can say this is the backbone. It is made up of phosphate, or you can say deoxyribose sugar, deoxyribose sugar and phosphate group. And in the center, there are nitrogen bases. The nitrogen, four types of nitrogen bases are adenine, cytosine, guanine, and thymine. Adenine, cytosine, guanine, and thymine, these are the four nitrogen bases which are present in these DNA strands. Under normal circumstances, the nitrogen containing base adenine always pair with the thymine. Adenine always pair with the thymine, and cytosine always pair with the guanine. So the binding of these pairs forms the structure of DNA. So they will uh, join together and there will be the formation of this DNA strand. And in the next, you can have a nucleotide. And you know, the DNA strand consists of, or the chain of DNA strand it consists of nucleotides. Many nucleotides, they will join together to make one strand of DNA. And one strand of DNA is made up of a phosphate molecule or pentose sugar or deoxyribose sugar, and then one nitrogen base, which may be adenine, thymine, cytosine, or guanine. So here you can see these two DNA strands in which base pairing is taking place. That is, adenine always combined with the thymine with the help of a double bond, whereas cytosine will join with the guanine with a triple bond. So same in the same way, the nitrogen bases will join together and they will give rise the large and long strands of DNA. And these two DNA strands are twisted together in this form, and they will make a double helical structure. So double helical structure, adenine always pairs with thymine and cytosine always pair with guanine. So in this way, there will be the pairing of these nitrogen bases with one another, and as a result, there will be long strands of DNA. Now, when we are discussing the DNA, the genetic code is a very specific word which is used in this regards. The genetic code is actually consists of triplets. Triplets, the group of three nucleotides that together encode one amino acid named a codon. So it means one amino acid was named a codon, in which three nucleotides are grouped together, as three nitrogen bases are involved in the formation of one amino acid. Each nucleotide, one of the four nitrogen bases, so a string of nucleotides hold a sequence of bases. This sequence form a code which instructs the cell to make specific proteins. As for example, C always pair with adenine, always pair with thymine, and cytosine always pair with guanine, and guanine with cytosine, and thymine with adenine. So in this pair, there will be the formation of the specific codes. DNA control all functions by controlling the production of proteins, including enzymes. You know, the enzymes are also the protein in nature. So enzyme control formation of all types of uh, proteins, and proteins are made from amino acids. 20 naturally occurring amino acids are there, which join together with the help of peptide bonds to make macromolecules or the large molecules of proteins. So these type of and sequence of the amino acid join together, which determines the kind of protein formed. You know that these 20 different amino acids are making the different types of proteins. So, for example, one protein molecule may start with sequence alanine, glycine, glycine, alanine, glycine, glycine. So this is one type of protein. And in the other type of protein, which can start with glycine, serine, alanine. So in this way, the 20 amino acids join together in differently to make or to produce the different types of proteins. And here you can see how genetic code will make a one amino acid. So here you can say the first letter, the second letter, and the third letter. When we will join the first letter, as in this case, first letter is U, and the second letter is also U, and the third letter is also U. So there will be the formation of one type of amino acid, and in the second U, and U, second letter and third will be C. So there is another type of nucleotide, and then U, U, A, and then U, U, G. So in this way, these genetic codes form the amino acids. So leucine, phelanine, leucine, and lysine, methionine, valine. So the different types of amino acids are formed with the help of this genetic code. And after discussing this genetic code, we can say that a gene is a sequence of triplets of 4 bases and which codes for a complete protein. Here we can example of insulin. Insulin is a protein, you know. Insulin is secreted by the pancreas to control the production of glucose. So the insulin is a small protein which only 51 amino acids. It means 51 amino acids join together to make insulin molecule, and each amino acid has 51 amino acids, and each amino acid has 3 nitrogen base. 51 is multiplied by 3 will give rise to the 153. A sequence of 153 bases in the DNA molecule would represent the gene that makes an islet cell in the pancreas. And would represent the gene that makes most proteins are much larger than this, and most genes contain 1,000 or more bases. So the proteins can be larger than 151, or they can have many large molecules inside. In this way, the genes determine the structure and function of the whole organism. So the different proteins are different in length and structure as well. So when they are different in structure and in length, they will perform the different functions. So they are different from one another, only the sequence of these amino acids. Other proteins coded for DNA include antibodies and membrane carriers and the receptors for neurotransmitters. So in this way, we have completed the part 1. Please like, subscribe, and share my channel. And thanks for watching my videos.